So we have just covered the structure of the ribosome and how the ribosome is able to catalyze the polypeptide, the formation of the polypeptide chain. So the next step is to look at the different steps of the translation process. So the first step that we have covered previously is the fact that the amino acids arrive charged to a tRNA molecule and that they are activated, so they are amino acid cells. Energy has already been spent and they're ready to react and form the peptide bond. So the first step of the process of translation is initiation. So the ribosome has to recognize the mRNA molecule. It has to assemble itself because it doesn't exist as a complete, both large and small subunit inside a cytosol unless it's bound to the mRNA. That's what allows the two subunits to come together. This is really important because it prevents random catalysis, random polypeptide formation just with the tRNAs but without a template. So the process of initiation, we're going to look over that first. Then there's the cyclical process of elongation. So the ribosome reads a codon, incorporates the amino acid and moves on, reads a codon, incorporates amino acid and moves on. There's initiation factors involved in the initiation of translation, and there's processivity and elongation factors involved in elongation of the polypeptide chain. Then the final step is a termination of the polypeptide formation. You'll remember that termination will occur at the stop codon when instead of an amino acid, a different type of molecule will be incorporated. Instead of the amino acid, nothing will be incorporated into the polypeptide chain, and instead the ribosome will fall apart and fall off the mRNA. And then, not in this set of lectures, but in the next set of lectures, we will also talk about protein structure and folding again specifically, and different types of post-translational regulation that can occur, as well as post-translational processing. This all depends on how much time we have. So here's the overview from your book showing how the process occurs. So the first, the small subunit is the first subunit that will bind and recognize the mRNA molecule. The first subunit will come already pre-charged with the methionine tRNA that will assist its recognition of the start codon. The 40S ribosomal subunit will attach at the 5' end of the mRNA molecule and then slide along until it finds the start codon. And only once it has found the start codon and the methionine tRNA is bound to the start codon will the 60S ribosomal subunit, the large ribosomal subunit, be able to recognize this formation and assemble the fully functional ribosome. This whole process is a process of initiation. It's first the 40S binds, then the 60S binds, then translation can occur. This is the process of elongation. So how is it that the tRNA molecules move between the three sites inside the ribosome, and how the polypeptide bond formation, how the peptide bond formation occurs inside the 60S catalytic large subunit of the ribosome. Finally, once the ribosome has been doing elongating for some time and it encounters the stop codon, there's termination factors come in. They cause the release of the polypeptide chain and then the disassembly of the 60S and 40S and mRNA triplets, causing the whole system to fall apart and dissociate. And that is the process of termination. Now, the process that we're not covering today, this time in lecture, but hopefully we will cover in the lecture on protein structure next, is the fact that the ribosome just makes the polypeptide chain. It strings the amino acids together. But a polypeptide chain is not fully functional until it's properly folded in the correct three-dimensional shape. And a lot of proteins require assistance with that process as well. So, translation initiation. Let's look at this picture first. So here you can see that multiple ribosomes can attach to mRNA message, and we're talking here prokaryotic transcription because you can see the DNA. The RNA polymerase is cranking out that mRNA, and the ribosomes are finding it right away and moving into this direction, reading the mRNA and translating it into protein. And you can see here an actual an actual process of the assembly. You can see that the 30S comes in first, then the 50S, and then the full ribosome has been assembled. So you can see again, simplified the process here. The first thing that comes in is the small ribosomal subunit and the initiator tRNA from methionine, and then only when these two are there on the starting codon sitting ready to go can the large catalytic subunit actually come in. So your book only covers the prokaryotic translation initiation, but I'm going to talk about the eukaryotic translation initiation as well. There's just more factors involved that I don't need you guys to remember. What I want you to realize is that the prokaryotic translation initiation is actually a little bit more complex than the eukaryotic, despite the fact that eukaryotes use a bunch more proteins. So, there's a particular sequence in front of the starting codon inside bacterial mRNAs called the Schindel-Garner sequence. It's named like that based on the discoverers of the sequence. It's this AGG, AGGU. The 16S RNA of the small ribosomal subunit recognizes that Schindel-Garner sequence and base pairs to it, allowing the smaller ribosomal subunit to stop at that location and pause enough for the tRNA with the starting methionine to arrive. The Schindel-Garner sequence helps to set the frame of translation in bacteria, because bacteria are notorious about putting multiple proteins onto the same mRNA message, something that eukaryotes rarely do. And here is the description of the prokaryotic translation initiation process. So there's a number of initiation factors that assist the small subunit in finding its mRNA and binding and positioning itself correctly on it. The first two guys are the initiation factor 3 and initiation factor 1. I'm not going to make you guys remember the numbers. You can if you'd like, but I think you can always look something like that up. But understanding why it is a 16S RNA that comes in there and what does that need inside the mRNA message in order to bind properly is important. Because if you ever are to try to make bacterial cell produce protein for you, you will need to make sure that your mRNA message doesn't just have a starting codon that's critical. You will not have a protein message, correct protein message, unless you have that starting codon, AUG. But if it has a good Schindel-Garner sequence in front of the AUG, so that the ribosomes bind to your mRNA message efficiently and translate it efficiently. So the initiation factors 3 and 1 bind first. The interaction with the initiation factors allows the mRNA allows the 16S subunit to read through the mRNA, and initiation factor 1 pauses the 16S ribosomal subunit at right at the AUG. It is the 16S rRNA that recognizes the Schindel-Garner sequence. You can see it right here. And the 16S rRNA is part of the small ribosomal subunit. Now, the initiation factor 2 is a GTPase that comes with it recognizes the starting methionine, the FMET of bacterial initiation, the methionine there is modified. It's F-meth, not just methionine. The binding between initiation factor 2 and initiation factor 1 positions the starting methionine tRNA perfectly into that AUG site, creating something that the large subunit of the ribosome can recognize easily. The GTP of the initiation factor 2 is hydrolyzed to form GDP. That triggers the 3 initiation factors to leave, and now the 50S ribosomal subunit can come in and form the fully functional ribosome, which is also called the 70S, which is just 50 plus 3. So this is prokaryotic translation initiation.